Hey everybody, we are beginning to really accelerate our readings and lecture schedules. Hopefully you all caught up with the information on Rosalind Franklin, as well as the videos and detailed written information from the Khan Academy site that I posted last week. I'm anxious to see what you think about the Khan Academy videos, as well as the supplemental materials that they have there. So you can always put a comment onto Piazza and let me know what you think about those. So we're really getting ready to talk a little bit about DNA replication. And of course, this is the first of our conversations about central dogma. And I'm sure all of you remember talking about central dogma from high school biology. As well as from Cells and Molecules at Stockton, or BIOL1 from your, if you're a transfer student coming to Stockton now. Central dogma, of course, the word dogma is one that means truth, and truth with a capital T. It was a little bit of an inside joke by Francis Crick, yes, of Watson and Crick. Crick was a noted atheist and in fact used the idea of dogma, which is usually meant to indicate a central tenet in different religions, kind of made a bit of a joke that central dogma in biology, this is the central truth, okay? For those of you movie buffs out there, also remember Dogma was a movie and in which Alanis Morissette played God. So there's kind of a, if you ever get tired during quarantine or if staying at home, take a peek, see if you can find Dogma and put it onto your watch list. But for Crick, it was really a tremendous insight in that DNA is an informational molecule. It contains information, okay? And he felt that he gave the idea that DNA was always involved in replication, and he said it's the only molecule that can replicate itself. And then, of course, DNA gets transcribed into RNA, and RNA gets translated into protein. So again, central dogma, three processes: replication, transcription, translation, and these are really the next set of three mini lectures. I don't spend a tremendous amount of time and effort from a molecular biology or biochemistry point of view on these topics, just enough for you to follow through and to be able to understand some of the advanced concepts that we're going to get to toward the end of this week and, of course, into next week, because we really need to be able to understand where genomes come from, a little bit about bioinformatics, genomics, etc. So we need to know enough molecular biology and biochemistry to get there, but I'm not going to be overly detailed because you haven't had, or at least most of you have not had, biochemistry yet. So, and in fact, some of you are taking concurrently organic chemistry. So we don't want to be overly detailed at this point in time. So the simplest of organisms, in which much of the original data was collected to talk about DNA replication, came from E. coli. As you know, E. coli is a prokaryote, has no membrane-bound organelles, a single circular chromosome, and of course the idea is, remember that DNA replication is indeed semi-conservative. It begins at a fixed origin, so you can see that little dot in the 10 o'clock position of the chromosome, and it is labeled as the origin. So that is where replication begins. We'll see a little more detail about how that starts shortly. Obviously, replication depends on DNA polymerase. DNA polymerase always moves from a 5' to 3' direction, okay? Always from a 5' to 3' direction in DNA. It requires a primer. The primer is usually an RNA-based primer, and it provides kind of a landing strip for the DNA polymerase to land and to begin to function. And synthesis of DNA proceeds bidirectionally. That is, it goes in both sides at the same time. So as the strands of DNA separate within the chromosome, replication occurs in both directions at the same time, and eventually leading to two new molecules, two daughter molecules of DNA. A little more detail on exactly how this works. We know that DNA is synthesized from individual deoxyribonucleoside triphosphates, or DNTPs. Think for a moment, how many different DNTPs do you think there are? Okay, obviously we've got adenine, thymine, guanine, cytosine. So there are actually 4 different DNTPs that function here. And so we've got these different DNTPs. Notice the position, because this is deoxyribose as the sugar in the nucleoside. Notice that we've got the 3rd carbon in ribose has the hydroxyl group, and then of course the 5th carbon up here is attached to where the phosphates exist. So again, remember 5' to 3'. These triphosphates are very important because you know of another molecule that is very, very similar and has the word triphosphate in it. Ever heard of ATP? Function of ATP? Correct. To provide energy in the cell. It is the source of chemical energy. So we've got ATP. Now we've got DNTPs. Okay? One of the advantages is we have these very active and energetic phosphate groups that help to provide energy for replication to occur. When we think about the beginning of replication, the first thing that happens is an enzyme called DNA helicase binds to the origin and begins to helicase. Remember, ASE is an enzyme. Helicase, it attaches to the double helix and begins to unwind the strand. Simultaneously, single-strand binding proteins attached to the single strand of DNA, and the purpose of that is that the DNA, as the helicase functions to unwind the double helix by breaking the hydrogen bonds, okay, the single-stranded DNA in the middle just doesn't reform. So we have these single-stranded binding proteins. Finally, another enzyme, DNA gyrase, prevents the remainder of the DNA molecule that's in the process of unwinding, prevents it from basically binding up and creating a tangle. So it relieves the stress on the double helix, creating what's known as a bubble. A bubble. Oops. And as this bubble begins here, we begin to see there is, in the green, we see a primer gets attached to the single strand of DNA by DNA primase. So it's an RNA primer. And again, as I said earlier, that primer provides a landing strip for the DNA polymerase. And the DNA polymerase moves in a 5' to 3' direction. Now, of course, because DNA is anti-parallel, and because it starts in the center of the bubble, the initial DNA replication functions in two different directions in a continuous fashion, moving 5' to 3', and this is called the leading strand. The leading strand. Well, what the heck happens to this other strand that's really in the 3' to 5' direction? Well, as the helicase continues to unwind the DNA molecule, primase sets down again, and it can go ahead and proceed in a 5' to 3' direction and create another strand. So first we have the leading strand, and then secondarily the lagging strand begins. Now, as you may have noticed by now, the lagging strand is discontinuous because it constantly, as the DNA unwinds, it constantly presents with a brand new empty single-stranded portion of DNA, which provides room for primase to attach, to put down another RNA primer, and then another copy of DNA polymerase zooms across. So the leading strand, of course, is discontinuous, or rather is a continuous. The leading strand is a continuous path of DNA synthesis, whereas the lagging strand is discontinuous. So we end up with these portions of double-stranded DNA with gaps. Okay? And these portions of double-stranded DNA, these short areas on the lagging strand, are known as Okazaki fragments, named for the person who discovered them. So I always like this summary slide because this really tells you everything that you need to know about DNA replication. Notice we've got our helicase moving in, unwinding single-stranded binding proteins, DNA polymerase moving in a 5' to 3' direction. It is continuous. Primase drops down the RNA primer. Polymerase then moves on the lagging strand in a discontinuous manner, creating these Okazaki fragments. And of course, here's topoisomerase, also called gyrase, so they have two different names for the same enzyme. And this is really everything you need to know, that I'd like you to know, about DNA replication. One question you may have is exactly what happens to the gaps. And what happens in the gaps occurs when a different DNA polymerase 1, replaces the RNA primer or the RNA nucleotides with DNA. And of course, this leaves a small space because the enzyme did not connect all of the adjacent nucleotides. And what that leads us to is the last enzyme in our series, which is, of course, DNA ligase. And DNA ligase seals the nick with a phosphodiester bond between the 5' phosphate group and the 3' hydroxy group. Remember, always moving in a 5' to 3' direction, even DNA ligase. And so you can follow along in more detail, take some time to look at the associated slides that we have, and also, obviously, the text. One of the things that we see that does happen, again, right out of the textbook, is there is this kind of looping effect that occurs during DNA replication, and basically it stops the new or novel de novo strand of DNA from getting caught up and tangled up. And so we see that we have 2 units of DNA polymerase 3 and this allows 5' and 3' to take place on both antiparallel strands, so kind of a classic way to look at it. I have these summary slides put together. It's really very simple. These are the key points that I'd like you to remember. DNA synthesis, of course, is continuous. The progeny strand, or the de novo strand, okay, that becomes the daughter chromosome, is extended overall in the 5' to 3' direction. Again, the names 5' and 3' come from the direction of the phosphodiester bond and on the carbons that exist in the ribose molecule, deoxyribose molecule. But it is discontinuous, of course, in the 3' to 5' or on the lagging strand. Remember that DNA chains are initiated by RNA primers. This becomes important a little bit later on. We're going to talk about PCR, polymerase chain reaction, and how it works. And of course, we have to have a primer because there has to be a landing strip in order to tell DNA polymerase where to land and where to function. All DNA polymerases have an absolute requirement for a free 3' hydroxy on this primer strand. And of course, again, I cannot emphasize this enough, all DNA synthesis occurs in the 5' to 3' direction. All of the enzymes that are involved in replication are assembled into a replisome. So that combination of enzymes, single-stranded binding proteins, and of course the parent and daughter DNA strands are called the replisomes, and the place the replisome acts is called the replication fork. And the fork moves along the parental DNA molecule, obviously creating new double-stranded DNA. Now, you may be asking yourself at this point in time, you know that everything we've talked about is got to do with prokaryotes. Well, we know that eukaryotes are different, and there are two big differences that we know between prokaryotic DNA and eukaryotic DNA. And that is the way that the chromosome is organized. Prokaryotic DNA, there is one circular chromosome. In eukaryotes, DNA is linear and there are multiple chromosomes. So we have a couple of different things going on that we need to talk about, just to talk a little bit more about it. First of all, how do we know that there is semi-conservative replication in eukaryotes, which of course are diploid? And how do we know that it is the same as the haploid circular chromosomes? Replication is the same in haploid circular chromosomes as in and is it the same in eukaryotes? And a great experiment was done here where chromosomes were labeled with tritiated thymidine, that is radioactive thymidine. The chromosomes were isolated during metaphase, and then of course you could see the theory is that both of the chromosomes or the sister chromatids will actually be radioactive, and then we put the cells into a medium without radioactive thymidine, so the new strand theoretically should be unlabeled. Okay, and you can see this in step 4. And here are the actual data. Very nice, showing you in the first metaphase, here are the autoradiographs, so the cells are separated at metaphase, placed onto a photographic plate, just like an X-ray, developed over time, and you could see how both of the sister chromatids are in fact radioactive. You see those little dots, the dark dots are the radioactivity. And then after the second metaphase, after they are replicated in a non-radioactive media, you could see half the chromosomes are totally de novo and they do not have radiation. There's a little bit of swap over, but that's probably some level of contamination. These are the original data, and this picture is from Taylor's work in the 1960s. So really very, very cool look at how we know that replication in eukaryotes is semi-conservative. Ultimately, you can see that in eukaryotes, we have an origin, we have a replication bubble, leading strand still functions in a 5' to 3' direction. The lagging strand still has Okazaki fragments. We still have helicase, we still have gyrase, we still have primase. All of these enzymes function exactly the same way in eukaryotes as prokaryotes. However, each chromosome has multiple sites of origin. And so all of these bubbles begin simultaneously, and the replication forks move toward one another during replication, and ultimately merging together. Of course, at the ends of the chromosomes, you can't really replace these primers. Why? Because there is no 3' hydroxy group. There's no free 3' hydroxy group. However, notice that all the replication fork, everybody's got both lagging and leading strands come together. Same thing happens in here, where we see polymerase 1 come in, fix the RNA, replace it with a deoxyribonucleotide, and then of course ligase comes in and seals the nicks. However, there is a gap left by the removal of the primer because of this, the non-existence of a 3' hydroxy. So there's no place to attach the novel bit. So theoretically, every time replication occurs, chromosomes should be getting shorter and shorter and shorter, creating a major problem. Of course, what we've got here is there is a novel molecule called telomerase. And telomerase basically has a sequence matching the same genetic sequence as the telomeres in the chromosome. It is an embedded bit of telomerase RNA. So telomerase is a hybrid molecule, that it is part protein and part RNA, and contains reverse transcriptase activity. So what it does is it attaches to the complementary sequence up at the top of the double-stranded DNA. Upon doing that, we now have a free 3' hydroxy group, and so we see that we can completely resynthesize DNA, and then of course remove the RNA primer, and the telomere is extended by a single repeat unit. Now, little difficult to understand just from this brief discussion. However, accompanying this short video is another video by Nobel Prize winner Elizabeth Blackburn. It is an excellent video, again, not too long, but it is her discussion of the discovery of telomerase and exactly how it functions, and you'll see how it complements the slides and the textbook, and she does a phenomenal job. I was lucky enough to be on a invited symposium along with Dr. Blackburn it was really a treat to be able to sit with her and chat with her. Again, not my main area of research, but it was a very varied symposium, a lot of different people, and great to have an opportunity to chat and learn more about her work. Really fun stuff, and she does a great job in the video too. Finally, this idea of telomeres, here we have Francis Collins in the background in the glasses. Francis Collins, of course, was head of the National Human Genome Project for quite some time and is currently the head and director of the National Institutes of Health, and had done some work showing that shorter telomeres are associated with cellular senescence and death. And this fella who is in the slide, apparently being aged, probably you might guess is in his 60s or 70s, is actually in his late teens here, and is showing, has a disease where he has premature shortening of the telomeres, promoting this. So this is one of the ideas, that telomere length can be associated with aging. So really some exciting stuff from the basic biology of understanding replication. So that's it for here. Hope you guys are doing well. Stay safe. Please remember to keep up with the readings in the textbook. This is the chapter on replication. And please also remember to watch the video with Elizabeth Blackburn. She does a stellar job. Take care. Talk to you soon.